Let's say we have a male who suffers from celiac disease. This man continues to negligently ingest bread, a gluten-containing food product which causes him to experience chronic diarrhea and atrophies his small intestinal villi. To prevent the further damage this man is knowingly doing to his small intestine, what innovative treatment will allow him to ingest gluten without experiencing the disease's complications? Firstly, I'll need to provide you with background information on celiac disease in order for you, a first-year medical science student, to understand my innovative treatment plan. Now, celiac disease is an autoimmune disease and is characterized as a chronic inflammatory disorder of the small intestine caused by the ingestion of wheat, rye, and barley. How does celiac disease manifest? Well, chemically within gluten, its protein gluten contains disulfide and hydrogen bonds to connect and stabilize gluten's polymers. In celiac sufferers, these chemical bonds cannot be broken and absorbed in the small intestine by the proteolytic enzyme chymotrypsin. Instead of chymotrypsin breaking down gluten proteins, an immune response is generated, whereby CD4 T lymphocytes identify gluten peptides as antigens. CD4 T cells then deamidates these glutamine residues in gluten by converting them to glutamic acid. Once converted, these deamidated peptides adhere to the HLA-DQ2 molecules within the lamina propria, thus provoking CD4 T lymphocytes to invade the lamina propria and produce inflammatory cytokines, which extirpates the small intestine's epithelial cells and flattens, atrophies the villi within the mucosa. With the only effective treatment for celiac disease being a strict lifelong gluten-free diet, I have developed a treatment method which possibly allows a negligent male celiac sufferer introduced at the beginning of this video to ingest gluten without complications. I have identified two specific species of bacteria that reside in an individual's oral cavity called Rothia mucilaginacea and Rothia area that can hydrolyze immunogenic gluten peptides in controlled experiments. However, these Rothia species don't possess the physiology to bypass through the hydrochloric acid in the stomach's acidic pH environment of 2.0, thus rendering the bacteria inactive and unable to reactivate once transferred to the duodenum in the small intestine. It was found that both species can thrive in hydrolyzed gluten peptides rapidly at a pH of 7.0 within controlled conditions. However, hydrolyzing rates steadily decrease above and below a pH of 7.0. Within the duodenum, the pH fluctuates between 5.0 and 6.0, which is not physiologically favorable for both Rothia species. Nonetheless, both bacteria can survive and still effectively hydrolyze gluten peptides within the small intestine. Now, I know what you're thinking. Won't the CD4 T lymphocytes generate a harmful immune response before the Rothia bacteria can completely hydrolyze gluten peptides? Well, you're right. If you extract glutamine endoprotease from barley, this protein can neutralize gluten's immunotoxicity. Therefore, by genetically modifying both Rothia mucilaginacea and area, incorporating glutamine endoprotease's genome into the bacteria's genome, then the genetically modified Rothia bacteria will then be able to neutralize gluten's immunotoxicity within the small intestine during hydrolysis, labeling gluten as a harmless biomolecule and preventing a harmful immune response. How will the Rothia bacteria reach the duodenum successfully? Well, the answer is microencapsulation. Microcapsules are made of alginate polymers which protect both Rothia species from the stomach's hydrochloric acid, increasing its survival rates within the stomach and allowing the Rothia bacteria to thrive in large colonies in the small intestine. Therefore, the genetically modified Rothia bacteria will then colonize the small intestine, suppress the immune response to gluten, and successfully hydrolyze gluten peptides. The continual ingestion of genetically modified Rothia bacteria is essential to maintaining large colonies which successfully hydrolyze and absorb gluten, thus allowing the negligent celiac sufferer mentioned at the beginning of this video to successfully ingest gluten without complications.